Hello everyone, let us continue with the chapter Molecular Basis of Inheritance Part 19. And last class we have studied about DNA fingerprinting, the introduction and the steps involved in DNA fingerprinting we have seen. And this class we are going to study about the schematic representation of DNA fingerprinting. And we have studied that the satellite DNA helps in identifying the differences among the individuals. So it shows very high degree of polymorphism, which forms the basis of what? Fingerprinting, right? Okay, this DNA fingerprinting, it is carried out in case of to identify the criminals and even in paternity disputes, right? In case, just for an example, okay, we have taken the DNA from individual A and the DNA from individual B. This is a schematic representation of DNA fingerprinting that is given in the NCERT textbook. Okay. And when you just look at this DNA, you can see here just to represent the chromosomes, right? It will be easy to learn. So that is why here it is being colored. So few representative chromosomes are being shown here where we have given chromosome 7, chromosome 2 and chromosome 16. So we have taken the same chromosomes in both individual A and individual B. So there are two persons, right? The DNA taken from two individuals, one is A, one is mentioned as B. And when you look at the chromosome, the same chromosome is been identified from both the individuals A and B, right? So this is chromosome 7. When you just look at these chromosomes, normally they are homologous chromosomes, right? Okay. Okay, so when you look at here, so in human we have studied that 46 number of chromosomes are present. So 23 comes from the father and 23 comes from the mother. Okay, so when you just look at the homologous pair here, one is the paternal chromosome, the other one is the maternal chromosome. So one which has come from the mother, the other which has come from the father. Right, so that is why here we have mentioned here. So this is chromosome 7. Okay, likewise when you just take here individual B, one is from paternal chromosome and one is from maternal chromosome. Mother and the father, right? Okay, so we have taken two different individual, right? And when you just look at this, what does this represents? VNTR, variable number of tandem repeats. Variable number of tandem repeats. So what is this tandem? Which is being repeated many times. So it will not be the same in each and every individual. It will be different. You can the genomic sequences. Here if you see it is 4. Okay, so likewise it varies. It is not the same in all the individuals. It will vary. So that number only you call that to be what? Tandem. Okay, so variable number of tandem repeats. So where are these variable number of tandem repeats? Found in satellite DNA. That is in mini satellite. That is why we have learned that it shows very high degree of polymorphism. The polymorphism which occurs due to mutation, the variation at genetic level. So with the help of this, we are going to find out who is the criminal. So what do they do normally? They have taken the sample from the criminal. Okay, the same pattern will be identified and they are going to check whether individual A matches or whether individual B matches. Okay, so what are the steps involved in DNA fingerprinting? We have studied isolation and then digestion and then separation of DNA fragments with the help of gel electrophoresis. What is that methodology called as agarose gel electrophoresis? When you arrange it according to the number of tandem repeats, example if you take 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, it moves on. Right? If you have the criminal, the same pattern of criminal sample, so what happens here according to the gel electrophoresis, what is being amplified, you are going to make it, this will be ready, right, to identify who is the criminal. Okay, so when you just check it here, what happens? See, if you count here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Right? So here you can see that. Okay. Likewise 4, 5, 6, 7, 8, 9. And here 6, 7, 8. And here 7. And here 5. And here it is 2. Okay. In case you want to identify the criminal, so you have to check now whose sample matches this criminal sample. Okay, right? So now we are going to arrange this pattern here in both A and B individual, and you're going to check which one matches this criminal sample. Clear? Okay, now if you look at this, the DNA from individual A with the pattern, okay, with the banding pattern where it is being shown here, and this is from individual A, okay, this is individual A and this is individual result of this, the criminal, what is being shown here, and this is the banding pattern of the individual A and this is the banding pattern of individual B. Okay, now we have to find out which banding pattern, okay, either it is individual A or it is individual B, which matches with the criminal sample. Okay, when you just compare with the criminal scene, which matches you can see here. So this is similar to this individual B. So the banding pattern of individual B matches the crime scene and it does not match with the individual A. So with the help of this DNA fingerprinting method, they will be able to find out the criminal. Next is applications of DNA fingerprinting. So when you look at this first application, it forms the basis of paternity testing in case of disputes. How? In case if there is a problem to find out whether the child belongs to this father or to the particular mother. In that case, they go in for DNA fingerprinting. Why? What did we learn in last class? Variable number of tandem repeats, where it is present in the satellite DNA, where it shows very high degree of polymorphism. And in this case, we have studied that even though it shows high degree of polymorphism, it is inheritable. They are non-coding sequences which does not code for anything. But still it is inheritable. They are transferred from the parents to the offspring. So, in schematic representation, we have studied that 50% comes from the father and 50% comes from the mother. That is why we have drawn that one is paternal chromosome, the other one is maternal chromosome. Because 23 comes from sperm and 23 comes from ovum. So it is equally shared. When the zygote receives, 23 chromosomes comes from the sperm and 23 chromosomes comes from the ovum. Both together you call that to be diploid. 46 in number. Diploid, fertilized egg, zygote is diploid. So the same way here also, the satellite DNA, which shows very high degree of polymorphism, it is inheritable from the parents to the offspring. So 50% comes from the father and 50% comes from the mother. When you look at the schematic representation, what we have studied now, how it matches individual A and individual B along with the criminal scene, right? The same way, the child's DNA sequence will be taken and then it will be compared to the mother's sample and the father's sample. And if you wanted to find out who is that father, so with the help of the DNA sample which is taken from that father, will be identified or will be checked with the child sample offspring. So in that case, if it matches, it is confirmed that that person will be the father. So how? With the help of this satellite DNA, because it shows high degree of polymorphism. So that is why here it is a basis of paternity testing in case of dispute, right? Disputed parentage, right? So next one, identify the criminal. We have studied how to identify the criminal, right? After completing the gel electrophoresis method, Agarose gel electrophoresis method, the DNA will be separated, right? All those procedures what we have studied. So using VNTR, variable number of tandem repeats as a probe, we are able to separate the fragments with the help of the autoradiogram, everything. Then you will be able to come to a conclusion who is that criminal, whether individual A matches with the criminal scene or individual B matches with the criminal scene. How? Even small blood sample which is found in the victim clothes, even after many years, even after several years, from the stain they will be able to identify the criminal. So where can you take this DNA sample? Right? From sperm, saliva, then blood, then skin, hair follicle, then bone. So all these are available. So very easily you will be able to identify, isolate the DNA. The same way from the victim cloth, even from the victim clothes, from the stain, right? So from that the DNA sample can be taken and you will be able to identify the criminal even after several years. Okay? Right. Next one, even in determining population and genetic diversity is also, this DNA fingerprinting has become very success. And one important thing what I have to know here is, in India, The first test of DNA fingerprinting was done in June 1989. For what? For setting a disputed parentage in Madras. So first in India it was done in Chennai, June 1989. For what? The same parental dispute, right? Clear? That is why. So who initially developed this DNA fingerprinting? Alec Jeffrey. That is why here Alec Jeffrey is called as father of DNA fingerprinting. Who is Alec Jeffrey? Father of DNA fingerprinting. Okay, so one important thing what I have to remember here is the satellite DNA, as it shows very high degree of polymorphism, it forms the basis of fingerprinting. If you like this video, share, comment and subscribe my YouTube channel and follow me on Instagram and Facebook. And the link is available in the description box. Thank you.